All right, y'all, so it's about time to start doing our cell culture. We got all our media and stuff thawed and ready. The biological safety cabinet is ready. The only thing I need to do now is take everything from on this side of the room and stick it in the biological safety cabinet. And so what I would tell you is I like to get everything I can ready before the fact. That way I don't have to do a lot of stuff in the biological safety cabinet and don't have to spend a lot of time going in and out of the biological safety cabinet. So I kind of already know my protocol that I had written down. I know what I'm going to do. I've planned for it. I've already labeled the tubes that I need so that that won't be a problem, right? That I won't have to worry about which tube is which in there. And that way I just, that's sort of stuff you can do out here on the bench rather than inside the cabinet. So let's go ahead and start moving stuff in there. I got my ethanol. I'm going to spray everything. Again, I'll start with spraying my hands. We're going to do as good a job as we can disinfecting. And for the tubes, I just want to spray them. I'll make sure they all surfaces. I'll just kind of throw them in there for now. Again, we're trying to not disrupt the airflow as much as possible. So just by throwing them in, we don't reach our hands in unnecessarily, but I am making sure all parts of the tubes get some ethanol on them. Just all over my hand. Okay, so that's good with the tubes. We're also going to put some PBS. We're not going to use it today, but I am going to aliquot some out, so we're going to go ahead and put that in there. Again, I spray it, and then I make sure all surfaces get covered and disinfected with PBS. Again, I'm going to move that off into the corner because we're not really going to use it. I know I'm going to do a couple flasks today. So the inside of this bag is not sterile, right? The outside of this flask is not sterile, but the inside of the flask is the important sterile thing. So we have to be careful that filter, right? That filter helps it breathe, but yet keep this sterile environment inside. So when I'm spraying, I'm being careful to cover the filter so I don't get any ethanol on that. And I can spray it down. I'll put that inside. And my second one to do the same thing. Again, you want to get some ethanol everywhere that you can without ruining that filter because you don't want any place for bacteria or viruses or anything else to grow. Okay, now I go to here where I can get my now melted bovine calf serum. Again, I'm going to spray that off really well. I'm going to put that in there. My pen strep, it's already aliquoted out. And finally, my DMEM. Okay, now I think we have everything we need in the biological safety cabinet. I'm going to go ahead and spray my hands off. And now I'm going to go in, and hopefully I will not need to come out of the cabinet for a while to get everything I need to be done. Okay, so I'm going to start organizing things here. So you see I have a place for sterile PBS. Those I'm going to use later. These ones that I have labeled for my media are there. And then I'm going to have some DMEM only where I'm going to take some of the DMEM out and put them in there according to the protocol. Okay, so first step here is to make media. Again, I know what I'm doing. I have my instructions over there in my cell culture menu. So I'm going to come out just so I can move that pen and see it. I've printed out instructions. I taped them in the lab notebook. Okay, I have everything I need. So to get started, I'm going to remove 100 mLs of this just DMEM only into these things. And so sometimes these are really hard to open and to find where the perforation is. Okay, so perforation's over here. I end up using the edge of this pen oftentimes once I do find the perforation. Get it open. This is my trash bin over here, so I can use that for trash. Okay, and so what I'm going to do first is I'm going to loosen some lids up. Right? I don't want to be struggling with lids when I'm holding sterile stuff in my hands. So remember, the inside of this is sterile. The outside is now sanitized. The inside of these tubes are sterile. Okay? We don't want to mess that up. And so remember, thinking about the airflow, thinking about where your hands are all really important. Okay, so in order to do this now, I'm going to use the pipetter. I'm going to use some 50 milliliter serological pipettes. So I'm going to go ahead and get a serological pipette on the pipetter. That's unfortunate. I got that. Okay, just put that to the side for now. Again, I'm going to have one flask opened here. I'm going to get 50 milliliters out. So I don't need to be exactly 50.000, but I want to be close. I'm going to close that. You're going to see it's dripping a little. You know, that's why we clean with ethanol. I'm going to open this in a way that I don't contaminate. Don't put my hands over the top of the opening. Put my 50 milliliters in there. That takes some dexterity to close, but you can close it. If you're really good, like I didn't contaminate that at all because I did not put my hand over top of that serological pipette, so I could still use it again. If for any reason you bump it or touch it to anything, it's no big deal, right? Just throw it away and try to, and if you don't know, just throw it away and use a different one. It's not worth contaminating cells when you don't know. So I'm going to get a second serological pipette just to show you it's no big deal. Get this out. Grab about 50, close. Again, this is you just get practice doing this one-handed. Put it in there. Okay, so we've removed the DMEM. Next step in the process is to add bovine calf serum. So I've already aliquoted these out at 45 microliters each, so I'm going to get another pipetter, serological pipette. Okay, and before I start to mess, I'm going to make sure I undo that a little bit. Again, it's okay if I have my hands underneath this, right? The problem is when I put my hands over top of something. So I open this up. I don't really care about what goes on inside that 50 ml tube after I get the bovine calf serum out. Okay, so now it's no big deal if I don't close it, but I'm going to close it anyway, just out of habit. You go ahead and put it inside here. All right, so we're done with the calf serum. And the last thing we need to add is 5 mils of pen strep. Again, I've already aliquoted out 5 mils, so I can use the 5 mil serological pipette. Hook it all up. So again, I don't really care what happens to this tube afterwards because we're throwing it out. This is a very drippy pipetter for some reason. That will happen. So I add my 5 mils. And we have our cell culture media. I'm going to go ahead and throw this in there. And this one in there as well. Okay, so now I will label and date, right? This now contains plus BCS, 10%, plus pen strep, 1%, and put my initials and my date. Okay, so what I wanted to do then is go ahead and aliquot out some 50 mil tubes. And the reason to do this is just so I don't have to get out this whole thing every time. And that way, if I end up accidentally contaminating some media, and I contaminate a 50 mil tube, okay, that's not great, but it's also not the end of the world and I don't contaminate my entire thing, right? So I'm going to try not to contaminate it, but just in case, you do have a little bit of a safety net by aliquoting some out into these 50 mil tubes. So again, I'm going to loosen, which I probably should have done before I even got the serological pipette out. I'm keeping the serological pipette up out of the way. I'm not touching anything with it. Go ahead and get 50 mils out. Being perfect on 50 mils here is not really important at all. I'm just trying to get some out. You know, one thing I could have considered doing here was like pipetting up and down a little bit to help mix all the stuff that I just put in here together. I think it mixes pretty well pretty quickly, so that's why I didn't necessarily do that. Okay, again, one thing open at a time. You see me just quickly close it. This is a very drippy pipetter. Okay, and so that time what I did is I went like this, right? And I put this back, and I noticed my hand went over the tip of this pipetter. And so that's why I paused. I'm like, oh crap, now I need a new one. And that's fine, right? I don't want to take the risk, so I'm going to go ahead and put this one away. And this is what I'm talking about always paying attention to where your hands are, what your hands are doing. You start to recognize the way things get contaminated in your cell culture. Okay, so next one. Okay, so this time I'm definitely keeping the pipetter tip out of the way of my hands, so I'm beside it. Finally, one last one at 50 mils. Close that up. Okay, so now I've made my media. I'm going to go ahead and close these up. These are all ready to go. The last thing I have to do is aliquot out me some sterile PBS. Same process. I'm going to get this out of the way and bring the PBS over here. I'm using 1X PBS. It is sterile, as long as one of your fellow students didn't mess it up. You're also noticing I'm trying not to rest my arms on these vents, blocking the airflow. Again, I don't need to get perfectly 50, so I don't need to spend a bunch of time being perfect about getting up and down. I just want some PBS to use in the future. I'm being careful on that pipette tip not to put my hand over it. Okay, good. So that's the first part of today's lab.